I want to start this deep dive with a story that has really stuck with me since we started digging into this. It's about a boy named Sam Berns. Now, you might have seen his TED Talk or heard his name. Sam was, incredibly bright, just so charismatic and full of life. But physically, visually, he was living a paradox.
It's a heartbreaking case, but it's also scientifically just profound. Sam died when he was only 17 years old.
17.
But if you looked at him, you didn't see a teenager. You saw the physical characteristics of an 80-year-old man.
Exactly. Balding, wrinkled skin, cardiovascular issues, diseases of the elderly happening to a high schooler. He had progeria, a condition of rapid aging.
Right.
And for the longest time, the medical community was just scratching its head. We usually think of aging as this slow, inevitable wear and tear, right?
Like a car that's just run too many miles. The assumption was that it must be some complex metabolic breakdown.
Maybe his metabolism was running too fast or something.
Or maybe his hormones were out of whack, something systemic.
But here is where it gets absolutely wild. It wasn't a metabolic disease. It wasn't a virus. It wasn't organ failure. The research shows it was a single mutation.
Just one.
One tiny typo in his DNA. And that typo wasn't in a gene for time or metabolism. It was in the code for a structural protein, a microscopic building block.
It really forces you to reframe how you think about biology. Sam's condition wasn't a software problem. It was a hardware problem.
A hardware problem. That's a great way to put it.
And to understand what happened to him, and to understand what is literally holding your own body together as you listen to this, we have to zoom past the organs, past the cells, and look at the steel cables of life.
We're talking about the intermediate filaments.
That's right.
So that is our mission for this deep dive. We are looking at the unsung heroes of the cell. We spent time in other deep dives talking about the movers, the proteins that carry cargo or pull muscles. But today
is about the protectors.
We're analyzing a lecture on cell cytoskeleton biochemistry to understand exactly how these filaments are built, why they are so incredibly strong, and, what happens when they break.
Because as Sam's story shows us, when these protectors fail, the consequences are catastrophic.
Okay, let's unpack this. We're calling them steel cables, but obviously they aren't made of metal. So what are we actually looking at here? If I shrink down to the size of a molecule, what do I see?
Fundamentally, intermediate filaments are fibrous proteins. They have what's called an alpha-helical structure.
Alpha-helical, so translate that for me.
Imagine a long coiled ribbon, or even better, think of a single strand of rope. But the magic isn't in that single strand. A single strand of anything is pretty weak.
Right.
The magic is in how they combine.
I found this part of the research really cool because the source material uses a story that I think everyone's heard. The fable of the old man and the bundle of sticks.
It's the perfect analogy for biochemistry. I love it.
Yeah, the father gives his sons a single stick and says, break it.
Snap. It breaks immediately. Then he bundles a dozen sticks together and says, okay, try now. And they can't do it.
Unity makes strength.
Exactly. That is the design philosophy of intermediate filaments. But, biologically, you can't just throw proteins in a pile and hope they stick together. The specific way they bundle is fascinating.
So walk us through the construction site. How do we get from one weak stick to this unbreakable cable?
It's a 4-step process. So first, you take 2 of those alpha-helical chains or single sticks, and you twist them around each other. That forms a dimer.
Okay, so we have a twisted pair, like a 2-strand braid.
Right. Then 2 of these dimers come together to form a tetramer. Tetra meaning 4. So now we have 4 strands.
Okay.
And this is the critical part that the source emphasizes. It's how they line up that matters. They align in an antiparallel fashion.
Antiparallel. I hear that and I'm picturing like traffic on a highway, cars going in opposite directions.
That is actually a great way to visualize it. In proteins, we have a head end, the amino end, and a tail end, the carboxyl end. Right. In this structure, the head of one dimer is sitting right next to the tail of the other. They're facing opposite ways.
Okay, but why? Why go to all that trouble? Is it just for symmetry? Or is there a function to it?
Oh, it's all about polarity. This is a huge concept in some ways forward.
So little delivery truck protein knows, okay, go toward the plus end.
Precisely. It's a one-way street. But intermediate filaments, because they are anti-parallel head to tail to head, the charges cancel out. They become non-polar.
So they're just neutral, structurally identical at both ends.
Yep.
Wait, isn't that a bad thing? If they don't have a direction, how does anything move on them? It sounds like building a road without any signs.
That's the point. They aren't roads, they are guardrails. If you're building a crash barrier on a highway, you don't care about the flow of traffic. You care about tensile strength. You want it to take a hit and not break.
I see. So because they don't have a flow, they are just pure static strength.
Well, static implies they do nothing, but stable is the word. Because they are nonpolar and they stagger themselves in these bundles, they are incredibly resistant to stretching. They form these massive coiled coils.
So they're not built for speed.
Not at all. They're not built for transport. They are built to absorb stress.
They are the shock absorbers of the cellular world.
Precisely. Once you have those tetramers, they just stack up side by side and end to end into these long rope-like structures. And that gives the cell its mechanical integrity.
Okay, so we've got the blueprint. Non-polar, antiparallel, super strong ropes. But looking at the source material, these aren't just generic ropes found everywhere, right? It looks like there are different models for different needs.
Correct. We classify them into 6 major types. Think of it like a hardware store. You have different cables for different jobs. You wouldn't use the same cable to hold up a bridge that you use to wire your headphones.
Right. So let's go on a tour of the body. Where do we find these things? Let's start with type I and type 2, the keratins.
Ah, keratins. You probably know these from shampoo commercials.
Strengthens your hair from root to tip.
For once, the marketing isn't really lying. Keratins are the intermediate filaments found in epithelial cells. So your skin, your hair, your nails.
And what are they actually doing in a skin cell? Are they just like filling space?
No. They're forming a structural network. Imagine a web stretching from the nucleus of the cell all the way out to the cell membrane. And crucially,
yes,
they connect to the filaments in the neighboring cells.
Oh, like stitching a quilt together.
Exactly. They act like rivets or stitches. Their main job is to protect against mechanical stress, friction, rubbing, stretching.
Which for skin is basically its whole job. I mean, it's the barrier between us and the world.
Right. If you didn't have keratin, or if you had a mutation in it, your skin cells wouldn't have that internal skeleton. The moment you rubbed your arm, the cells would literally tear apart.
Wow.
You get massive blistering from the slightest touch.
That is a terrifying image. It really highlights how much we take that toughness for granted. Okay, let's move deeper. Type 3. The source calls this the support crew. We've got vimentin and desmin.
Right. Let's start with vimentin.
Vimentin, what's its deal?
Vimentin is the most widely distributed. It's found in tissues that are dynamic, connective tissue, blood vessels.
The source mentions something specific about vimentin and organelles. It's not just holding the cell shape, is it?
No, it's like the internal scaffolding for the furniture. It positions the organelles. Think about the mitochondria, the power plants of the cell.
Okay.
They don't just float around in a soup. Vimentin helps anchor them in the right position so they can deliver energy efficiently.
Okay, but the other type 3 filament, desmin, this is the one that really stood out to me. The source uses a car analogy here that I thought was brilliant.
The car chassis analogy, I love this one. So desmin is found in muscle cells. Skeletal muscle, cardiac muscle.
Okay, so we're in the gym now.
We're in the gym. Now, to understand desmin, you have to understand how a muscle works. You have proteins called actin and myosin. These are the engines.
Right, they slide past each other to generate force.
Exactly. Engine creates the horsepower. Yeah. But imagine you take a Ferrari engine, incredible power, thousands of horsepower, and you just sit it on the asphalt. You rev it. What happens?
It makes a lot of noise, maybe shakes around, but it doesn't go anywhere.
Precisely. To move the car, you need a chassis. You need a frame to hold the engine and transfer that power to the wheels. Desmin is the chassis.
So desmin physically connects the engine to the cell structure.
It sits at the Z discs, which are like the anchor points in the muscle fiber, and it ties everything together. It ensures that when actin and myosin contract, the whole cell contracts uniformly.
This implies something really interesting about muscle diseases. You could have a good engine, your actin and myosin are fine, but if you have bad desmin,
you have a breaking car. The engine revs, but the force isn't distributed. The muscle is weak, or worse, the cell can tear itself apart under the force of its own contraction.
The structural failure again.
Exactly. It's not a fuel problem, it's a frame problem.
Oh, wow. Okay, moving on to type 4. Neurofilaments. These are in our neurons. What's their specific job?
This is all about the need for speed. Neurofilaments determine the diameter, the caliber of the axon.
The axon, that's the long wire that sends the signal from your spine to your toe.
That's the one. And diameter matters.
Why does it matter?
Physics. Think of it like a water pipe or an electrical wire. A wider cable offers less resistance.
So it conducts signals faster.
Much faster. So neurofilaments are basically responsible for upgrading your nervous system from dial-up to fiber optic.
That's a great way to think about it.
In a way, yes. They bulk up the axon to ensure nerve impulses travel at the correct speed. If you lose that structure, the wire gets too thin and the signal slows way down.
It's amazing how specialized these get. We skipped type 6, nestin, which is mostly for development, but I want to circle back to the one we left for last, type V.
The lamins.
This brings us back to the hook of our deep dive, back to Sam Berns. We talked about keratins in the skin, desmin in the muscle, but lamins are different. They aren't floating in the cytoplasm, are they?
No. Type V filaments, the lamins, have a VIP section. They are found exclusively inside the nucleus.
The command center.
Right. They form this dense meshwork called the nuclear lamina that lines the inner nuclear membrane.
I was picturing this like a hard shell, kind of like an eggshell inside the cell.
It's a bit more flexible than an eggshell, more like a stiff wire mesh lined the inside of a balloon. It keeps the nucleus spherical. But here is the deep dive detail that is crucial for understanding progeria.
Okay.
The lamins don't just hold the shape of the bag. They anchor the DNA itself.
They grab the DNA.
They act like a filing cabinet. Specifically, they anchor the heterochromatin. That's the silent, tightly packed DNA that the cell isn't using right now.
Oh, okay.
They pin it to the wall of the nucleus to keep it organized and out of the way.
So it's organization. You have the active files on the desk and the inactive files safely locked in the cabinet.
Exactly. Now, let's look at Sam Berns again. Progeria is caused by a mutation in the lamin A gene. This produces a defective protein called progerin.
And what does progerin do to our filing cabinet?
It makes it structurally unstable. The cabinet collapses. If you look at the nucleus of a cell with progeria under a microscope, it's not a nice round sphere. It's lobulated.
What does that mean?
It looks like a crushed ping pong ball.
And because the cabinet is crushed, the files are everywhere.
The organization of the genome is lost. The DNA that should be anchored to the wall comes loose. Genes that should be off might turn on. Genes that should be on might get damaged. The genome becomes unstable.
And when the genome is unstable, the cell, it panics.
It stops dividing. It goes into senescence. It effectively ages instantly.
This is the aha moment for me. Sam's accelerated aging wasn't because his clock was running fast. It was because the physical structure holding his genetic code safe collapsed.
It redefines what aging is. We usually think of aging as running out of time. But this suggests that at a cellular level, aging is structural fatigue.
It's the failure of the meshwork.
The failure of the meshwork that protects your blueprint.
Yeah.
It's a mechanical failure.
It is. And it shows us that you can't separate structure from function. You can have the best DNA in the world, but if the box you keep it in falls apart, the DNA gets damaged.
Exactly.
So let's zoom out. We've covered a lot of ground skin, muscle, nerves, the nucleus. If our listener is, say, a med student or just someone trying to organize this in their brain, how do we distinguish intermediate filament problems from other cell issues? The source gives a sort of clinical cheat sheet.
It's a great way to simplify it. It comes down to the primary deficit. If you have a problem with actin or microtubules, you usually see a problem with movement. A white blood cell can't crawl toward a bacteria. Sperm can't swim. Cilia can't beat.
So movement failure equals actin or microtubules.
But if you see tissue fragility, that is the key word. If the skin blisters because it can't handle friction. If the muscle is weak, despite having a working engine. If the nucleus collapses,
that's a structural failure.
That is an intermediate filament problem.
The structure is failing, not the motor.
Exactly. The engine runs, but the chassis is broken. That is the hallmark.
You know, we started this deep dive talking about unsung heroes, and I feel like that is the perfect description. You don't notice the steel beams in a skyscraper until the building starts to sway in the wind.
That's right. These proteins are doing their job silently right now as you sit there. They are resisting tension, absorbing stress, keeping your DNA organized.
They're the reason we don't just turn into a puddle of jelly.
Pretty much, yeah.
It really makes you think about the fragility of life. We walk around feeling solid, feeling strong, but really we are just a collection of fragile compartments held together by these tiny microscopic ropes.
And one tiny mutation, one little twist in that bundle of sticks, and the whole architecture can change.
It does. It reminds us that biology is, at its core, physical engineering. We are built structures. And understanding that architecture, how the beams are bolted together, is the first step to fixing it when it breaks.
So here's my challenge to you, the listener, as you go about your day. Flex a muscle. Pinch the skin on the back of your hand. Don't just feel the movement. Try to visualize those billions of microscopic steel cables, the keratins, the desmins, that are physically holding that tissue together against the stress.
It's a marvel of engineering happening right under your fingertips.
Thanks for diving deep with us today into the steel cables of the cell.
Always a pleasure.
We'll catch you on the next deep dive. Stay curious.